So, good afternoon, guys. So, we'll start with week 8 of the host pathogen interaction by Professor Himanshu Kumar. So, in this lecture, we'll be mostly discussing about the antigens as we discussed in the last class and the bit about the antibodies. Okay, so let's start the lecture starting with a brief introduction about the antigens. Okay, so antigen that can easily bind any agent that has the capability of binding specifically to the T cell receptor or any antibody, and the ability of binding to either of them is called as antigenicity. Okay, and there is another term that is immunogen. So that is any agent that is capable of inducing an immune response that is called as an immunogen, and the property is called as an immunogenicity. Okay, all immunogens are antigens, and all antigens are not immunogens. Okay, so all antigens do not elicit an immune response. So this we have to remember thoroughly. Okay, so what are the major properties of an antigen that makes it an antigen? Okay, so the first thing is foreignness. That means the antigen must be foreign to the body. So foreign means that must be unknown to the body. The all the proteins that are present in the body are known to the body, so they are not foreign. That's why they do not elicit any immune response. So the antigen must be foreign. So for example, BSA that is not immunogenic when injected into cow because it has been isolated from cow itself, or it can be less immunogenic in goat, but uh, when it is injected into chicken, then it will be more immunogenic because it is from a not a near it is not from a near relative of that particular organism, so that's why it will be foreign in chicken. Okay, so and some of the molecules that are highly conserved in nature throughout the evolution that elicit very little immunogenicity. Okay, so because if we talk about we have cytochrome C here, and we have the collagen. So any of these molecules, if we see for a rat and for a human, so we would see very minute differences in this case. So if we inject the collagen of rat to human, so in that case, what will happen? We would elicit minimum in immunogenicity. Okay, and but there are some exceptions to that. Why? Because the corneal tissue, for example, doesn't have any blood vessels. So the region is actually being not recognized by the body. So if we change it, the immune response will not be elicited without anything. Okay, and the sperm. So if we actually have a contact of this corneal tissue to the body, so that will elicit the immune response, although it can be from the same organism itself. Then we have another thing that is sperm. So sperm also, if we inject it into intravenously into the body, so it can elicit an immune response because it will not be in contact with the blood supply of that particular person. Okay, so these are the few exceptions in which the corneal tissue as well as the sperm can elicit the immune response in the same individual itself, while the cytochrome C and collagen cannot elicit an immune response in different individuals also, while the second property includes the size. So, for example, very small size molecules like antibiotics, so they are not immunogenic. But and 1000 to 6000 Dalton hormones, they may or may not be immunogenic. Okay, and greater than 6000 Dalton proteins that are albumin or toxins, they are immunogenic. So, as we can consider here, that between, so less than 1000 daltons of the proteins will not be immunogenic to the body, and greater than 6000 dalton will be immunogenic to the body. Okay, so moving on to the different proteins, as we can see here. So one we have BCG, that is bovine gamma globin, BGG, that is 150 kilodaltons. Okay. Then we have BSA 69 kilodalton, flagellin, that is a monomer 40,000 40 dalton, 40 kilodalton. Then we have hen egg white lysozyme 15 kilodalton. So these are the different weights of the proteins that can elicit an immune response into different organisms. Okay. Now we are moving on to the use of adjuvants to enhance immunogenicity. Okay, so the immunogenicity enhancing agents are called as adjuvants. So these are basically mm, the immune enhancing agents. Okay, so the mechanisms how an adjuvant actually induce an immunogenicity are not precisely clear, but they might activate the pathogen recognition receptors. Okay, so we have one type of adjuvant, that is water in oil. So mm, and the complete adjuvant, that means antigen in aqueous solution or mineral oil and the emulsifying agent. So these are the antigens that actually enhance the immunogenicity of the particular agent. Okay, antigen after covering from these solutions will be released slowly from the injection site. Okay, and then we have the second one, that is fruit Prudent's complete adjuvant. That means they have the heat killed mycobacteria. Okay, so the incomplete ones are basically the emulsifying agents that will help in slow release of the particular antigen, while the complete adjuvant will be the that would contain the heat killed mycobacteria. Okay, so that would actually enhance the immunogenicity. Okay, so there are certain adjuvants that we can see here. So these actually and their mode of Now we have the IgG, that is the most abundant antibody. It has a hinge region, and the classical example of an antibody that we study is the IgG. So this antibody has four subclasses. It can cross placenta, and it is also involved in the complement system, and it is 80% of the total IgG. So, in case of IgM, sometimes it is also referred to as a part of the innate immune system because it is part of the primary response, primary immune response, while IgG is totally part of the adaptive immune response. Okay, so there are subclasses based on the hinge region uh, on of them. So the Ig3 is basically the longest hinge region that the antibodies have, while the second highest is Ig2, while the third highest is Ig1 and Ig4. Okay, moving on to the IgA, that is the secretory antibody. So it is a dimer. Okay, so it exists as a dimer, and it can cross-link large antigens as well. Okay, then it is mainly present in the mucous membranes. It also has a J chain that is act as a connection between the two monomers of the antibodies. While because it is secreted in the mucous layer, it has a secretory component also that actually protects this particular chain or the antibodies from the protease action. Okay, so, and because it is delivered in a mucous membrane, so the secretory component becomes a necessity. Okay, moving on to the particular IgG secretion. So what happens? This is a plasma cell. Okay, so as you would remember, so B cell, when it will get a signal to produce the antibodies, it will divide into clonal population into memory B cell that will retain the memory of the antigen, and the plasma cell. So plasma cell will basically secrete the antibodies. So this plasma cell secretes the dimeric IgA. Now this dimeric IgA will bind to a poly Ig receptor that is present on the epithelial cells of the gut. So after this, it will engulf this antibody along with the receptor, and it will be in a vesicle. Okay. So now this vesicle will contain a secretory component to the IgG, and after the enzymatic cleavage, this receptor will get bound to the secretory IgA, and it will be released into the lumen. So this is called as transcytosis, that is only seen in case of the IgA secretion, where it binds to one end of the epithelial cell and gets secreted on the other end inside the gut. Okay, so this is a unique phenomena that is observed only in the secretion of the IgA. Then we have the last class, that is IgE. So this antibody is involved in the allergic reactions, and it is involved in the parasitic infections where it will be released, and it directly targets the parasites, and it directs the mast cells and the basophils to release the inflammatory molecules like histamine and serotonin. That is a key component in the allergic reactions. Okay, now we'll move on to the antibody gene arrangement. Okay, so what happens is whenever there is an antibody production, so the gene for the heavy and the light chain is same, single. So there would be some chances of generating the diversity of the antibody. Okay, so that we'll be going to see now. So, in germline DNA, there are multiple gene sequences that code for portions of the that code for portion of a single immunoglobulin heavy or light chains. Okay, so during maturation and stimulation, these gene segments are shuffled, leaving the coding sequence of only one functional heavy and light chain. Okay, so as we have just now discussed that there are multiple heavy or light chain. So in that case, what happens during maturation and stimulation, these heavy and light chains are transformed into only one functional thing that is expressed in the form of immunoglobulin. Okay, so the chromosomal DNA in a mature B cell or a plasma cell is will not be same as in the case of the germline DNA. So as we can see here, this is the germline DNA, and after the maturation, there are some segments that have been deleted, and this is the rearranged gene segment. So when we will actually sudden blot it, or we'll have a restriction enzyme digestion, then both of them will produce different type of fragments, because here we have the various germline various restriction enzyme sites, while in this case we have very minimal enzyme sites. So here we'll see a different band pattern as compared to the germline. Okay, so this is how we differentiate between the rearranged and the original germline DNA of the matured B cell. Okay, now moving on to the basic the different orientation and the cells that have different types of receptors. So the B cell are basically have a precursor called as hematopoietic stem cell. Okay, then it matures into lymphoid cell, followed by the partial heavy chain gene arrangement. Then it forms a pro B cell. Okay, so this all happens in a bone marrow. In this case also, then after that completing the heavy chain gene arrangement, it completes the pre-B cell formation. Then in the pre-B cell, the cells actually express the mu heavy chain with the surrogate light chain. Okay, so that will be a, not the actual side chain, but a surrogate side chain. Then in the pre-B cell, we have the light chain arrangement. So after that, it forms a immature B cell that have a membrane-bound IgM. Followed by the change in RNA processing, we have a mature B cell in which membrane-bound IgM and membrane-bound IgD are there. After that, the antigenic stimulation happens and we have the activated B cell. Followed by its differentiation, the IgM secreting plasma cells are formed that secrete the IgM. Now, after that, after this differentiation, the class switching happens and it forms either the memory B cells or it forms the plasma cell secreting IgG, IgA, or IgE. Okay, so this is how a B cell is formed, that is basically a plasma B cell. Okay, so till the maturation of the B cell, it is happening in the bone marrow, and after that, it happens in the peripheral lymphoid organs. Okay, so so we have the genetic organization of immunoglobulin germline gene segments. So this is the lambda chain DNA in which we have the uh, leader peptide L. Then we have the variable region of the variable region 2. Then J is the joining chain, and C is the constant region. Okay, so this is how we have different regions in the uh, segment. Okay, and this is the kappa chain, and in this case what happens, the n is nearly 85. Okay, so we have nearly 85 regions here. Okay, and in the heavy chain DNA, there are 134 domains that are present here. Okay, as we can see here. Although the lambda light chain is present on chromosome 22 and on mouse it is present on chromosome 16, the kappa light chain is present on chromosome 2 in humans and 6 in mouse, and the heavy chain is present in chromosome 14 in humans and 12 on mouse. So moving on to the heavy chain arrangement, as we have seen, the germline DNA is this one. Okay, so we have around variable regions of 134 here. Okay, then we have the D region as 13, then we have the J joining region, and then the constant chains. So this these constant chains make it a different types of isotypes of the particular uh, uh, antibodies. Then first we have the joining. So in this case, for example, any of the D can be joined to the J's. So the first the DJ joins, and any of these D's will join with the J. So this is called as the splicing reaction. Okay, so by splicing they join is like this. So all the D's that lies between the D6 will be maintained in the will also be spliced after the V joining to the DJ. Okay, now happens the V joining to the DJ. So the V actually have the sorry, D stands for diversity and J stands for the joining region. Okay, so now when this V needs to be connected to this DJ, so in that case what happens, there is removal of this in between segment through splicing, and hence this VDJ recombination takes place. Okay, now after that this transcription happens. So this after this leader peptide and this PDJ are formed, followed by the formation of the either uh, first the M mu and the delta are formed. Okay, so now it forms two types of transcript, one with the mu and one with the delta. So the rest of the exons are removed by splicing. So this is a signaling mechanism that the splicing takes place. Okay, so it forms the mu heavy chain, and this delta forms the delta heavy chain, and these are the first antibodies or the immunoglobulins that are attached to the membrane of the immune B cell. Okay, so this is what a mature B cell would look like after the heavy chain rearrangement. Okay, now moving on to the light chain gene arrangement here, the N is nearly 84. So the variable region can be multiplied to 84. That's why it is called as a variable region. So for in this, the D region is not there. So we have the VJ recombination here. Okay. So first the V recombines with the J and forming the VJ combined thing with which are preceded by the leader sequence. Now there is a gene arrangement with the rearranged kappa chain followed by the primary RNA transcript that have the VJ recombined, and then after the splicing it forms the VJ. And the constant region of the kappa chain, followed by its nascent peptide that have the VJ combined with the constant region of the kappa chain, and the light chain that codes for the variable and the constant region for the kappa chain. Okay, so this is how the light chain of the kappa chain is rearranged. Now moving on to the generation of the antibody diversity. So what happens? There are multiple germline gene segments. Okay. So the combinatorial VDJ joining by the VDJ recombinase, that is recombination activating genes 1 and 2, and the terminal deoxynucleotide transferases. So these actually generate the diversity, okay, for different binding of the antigens, and they have the junctional flexibility, and the P region of the nucleotide addition via the recombination signal sequences, the N region of the nucleotide addition via RSS, the somatic hypermutations, and the combinatorial association of the light and the heavy chains provide this generation of the antibody diversity. Okay, so these are the few points through which we have the generated the antibody diversity. So ensuring only one chromosome expresses the antibody genes alleles exclusion. So the rearranged heavy light chains from only one chromosome are expressed out of the maternal or the paternal chromosome. Okay, so this rearrangement will lead to the maternal H chain or the maternal kappa chain, while this is the maternal H chain or the. So this is how it maintains the diversity of the particular antibodies. Particular antibodies, yes. Okay. So now moving on to the junctional flexibility in joint. So here we have the nucleotides. Okay. So the V region when joins with the J region, so it leads to the joining flexibility between different nucleotides. Okay, so there are some productive rearrangements that takes place that leads to the formation of a functional chain, and there are some non-productive rearrangements also. Okay, then we have the class switching. So the switch region is between the 2 to 10 kb. We have the recombinases to allow that. We have a switch factor that happens with the IL4, and we have activation induced cytidine deaminase AID or RNA editing enzymes that change from C to U. Okay, then we have the combinatorial antibody diversity in humans and mice. So how they are generated based on the number of VDJ segments. Okay, so for the heavy chain we have V48, 23, and 6. Okay, so this is the number of segments in humans. And for the kappa and the delta chains, we have these number of segments. We don't have D region in the light chains. Okay, so when we multiply these, for the heavy chain we have around 6,000 combinations from the VDJ recombination, and for the kappa chain we have 205, and for the lambda we have 170 combinations. Okay, so the possible combinations between these three combined will be near to 10 to the power 6, that is 2 million. Okay, so that's how the antibody diversity is generated. Okay, now we'll be moving on to the hybridoma technology. So now we'll move to hybridoma technology, that is the production of monoclonal antibodies. Okay, so now what we'll do is we first have the mouse. So mouse is challenged with an antigen. Okay, so now these will have the spleen cells. Okay, now spleen cells will yield the antibodies. Then we have another type of cells that are myeloma cells. So myeloma cells are cancerous cells basically. So they will have a long lifespan. Okay, so after this, there if we fuse with them, so the spleen cells that are fused with the myeloma cells will called as hybrid cells or the hybridomas because of the myeloma. These hybridoma will now be cultured in a medium that is called as hat medium. So we have hypoxanthine aminopterin thymidine media. So to select the positive cells, that means the fusion of B cells with the myeloma cells will be taken into account, and then we'll use them to harvest the monoclonal antibodies. Okay, so now the first step is we'll immunize the mice with the and the selection of the mouse donors, so that we'll have a antibody titer in the serum. Okay, the first we'll immunize this mouse with the antigen, and then we'll see whether the antibody is being produced or not in the serum. Then accordingly we'll isolate the spleen to cultivate the B cells from this mice. Then after several weeks of immunization, we'll see the serum antibody titer through ELISA or flow cytometry. If the titer is too low, then we'll boost it with pure antigen. If the titer is high, after 2 weeks, we'll boost with the pure antigen. Okay, now Moving on to the third step, that is the preparation of the myeloma cells. Okay, so the myeloma cells are cancerous cells that have that doesn't have enzyme, that is HGPRT. Okay, so HGPRT is an enzyme that converts hypoxanthine to the nucleotides. Okay, so it converts it to the other nucleotides. Okay, so this doesn't have this HGPRT enzyme. That is why it is known as HDPRT negative, that have high viability and rapid growth. Okay, so these myeloma cells will now lead to now these are now what will happen, we'll grow them in the hat medium. So how we'll use this hat medium? So we have the aminopterin that will block the dihydrofolate to tetrahydrofolate conversion, that will help in the de novo synthesis of the nucleotides. While we have the HGPRT, so HGPRT will help in the salvage pathway. Okay, and the thymidine kinase will also help in the salvage pathway. So that's why the hat medium provides only the enzyme that uh, the cells that have the enzyme will grow in this medium, because the hypoxanthine will be converted, the thymidine will be converted, but aminopterin will block the de novo synthesis. Okay, so why this was done? Because when we add, so when we add the spleen cell with the myeloma cell, so we have different cell combination that can be seen after the fusion. Okay, so we have either so for example we have B cells here and we have myeloma cells here. So the particular combination after fusion that you can see will be either B cell to B cell fusion will be there, then M cell to M cell fusion will be there, or B cell to M cell fusion would be there. Okay, now, if we grow them in the hypoxanthine, aminopterin, and thymidine media, the myeloma cells doesn't have the HGPRT enzyme. So they will not be able to grow in this media, because in this case they don't have hypoxanthine, so HGPRT, so they'll not be able to take the salvage pathway, and aminopterin blocks the de novo pathway. So both the combination of M and M cells will be dead in that media. Now, if we see about the B cells, so B cell can be grown in this media because they have the HDPRT enzyme. So the B cell can grow in this media, but they have the limited lifespan. So these cells will be discarded because of the limited lifespan. So the ones that grow in this media are the fusion product of the B and the M cells, because they have a lifespan longer than the normal B cells, and they can grow in this media because they have HGPRT positive because of the B cells. Okay, so now we have selected these cells, and then we'll isolate the monoclonal antibodies from these particular cells. Okay, so moving on, then we then we expand these clones and test the supernatant for the antibodies. So we will expand only the positive clones. Okay, so what will be the main for purpose of producing this monoclonal and polyclonal antibodies is that in monoclonal antibodies basic research, they are immunoblot, immunohistochemistry, or cell analysis using cell cytometry. Further, in diagnosis, they can be used for pathogen detection, malignancies, and diagnosis of infection and autoimmune diseases. And in case of the therapeutics, they can be used in cancer, autoimmune, and allergy treatment. While in case of the polyclonal antibodies, they are used in immunoprecipitation, chip Immunoelectrophoresis or radio immunodiffusion. In diagnosis, they are used in histopathological analysis, and for the therapeutic use, they are used in passive immunization and anti. Okay, now we are moving to the T cell receptor. Now we are moving to the T cell receptor. So, like the B cell receptor, we have the T cell receptor also. So, the antigen specific nature of T cell responses clearly implies the presence of the T cell receptors. However, the TCR membrane bound and recognition is weaker than the BCR. That's why it requires the MHC complex to present the antigen. Then the complex cellular assays were required to reveal the presence. Okay, so as we can see here, for a cytotoxic T cell that is CD8 positive, we have MHC class 1 that actually being recognized by the CD8 receptors and that present to the TCR. For the CD4 T cells, the CD4 positive T cells or the T helper cells, the MHC class 2 actually present the antigen to the T cells. Okay, and here is the comparison between the T cell and the B cell receptor. So the B cell receptor, this is a membrane bound IgM that we have seen here. Okay, now yes, similarly, for the T cell receptor, we have the cytoplasmic tail, transmembrane domain, and a connective sequence. But here, the variable and the constant region is singular. Okay, as in case of the antibodies, we have a multiple constant region and the variable region. Here, it is singular, and this is the alpha beta T cell receptor. Okay, now we'll see on the CD3 complex. So this is the T cell receptor, and these are the different uh, receptors, that is gamma epsilon and epsilon delta receptor of the T cell that is also present on the uh, some T cells, and we have the ITM activation domain, that is immunoreceptor tyrosine based activation motif. Okay, so that helps in initiation of the signaling cascade. Okay, so here we have the accessory membrane molecules like CD4. It is present with the class 2 MHC molecule, help in adhesion and signal transduction. Similarly, CD8, the ligand is class 1 MHC, and it also helps in signal and the adhesion, signal transduction, and it is a member of the immunoglobulin superfamily. Okay, we have CD2 that binds to CD58, LFA1 that binds to ICAM1, CD28, B7, CTLA4, B7. So CD28 and B7 are co-stimulatory markers of the T cell, and CTLA4 and B7 are the inhibitory markers. Okay. Then we have CD45R, that is CD22, and CD5 is the receptor for the CD72 ligand. Okay. Now these are the different ligands and the receptors on the TH and the TEC cell. Just now, like what we have discussed. Okay. Moving on to now we'll move on to the questions. So, what role do MHC molecules play in the immune response? So, the options are: they directly kill the pathogens by proteolytic cleavage, they process and present antigens to T cells, they mediate antibody production, they activate B cells, presenting antigens to them, and they present nucleic acid antigens on their surface. So the correct option is option number 2. That means they process and present the antigens to the T cell, because they will not be able to, the T cells do not directly activate, bind to the antigens. Moving on to the second question, which of the following accurately describes IgM? So let's see the options. IgM is the first antibody produced in response to the antigen. Yes, that's why it is considered to be a part of the innate immune response. IgM exists only in monomeric form? No, it exists in both pentameric and monomeric forms. IgM has the highest complement activation potential. Yes, this is correct. IgM is the most abundant immunoglobulin in serum? No, it's IgG. IgM is present in monomeric form on mature B cells. Yes, this is correct. IgM can also be found in serum in the pentameric form. Yes. IgM has the highest half-life. Not really. And it does not bind to the complement proteins. No. So the correct answers are 1, 3, 5, and 6. Okay. So let's move on to the next question. In the context of immunoglobulin diversity, which enzymes are essential for initiating the VDJ recombination in B cells? So they are the RAG1 and RAG2, DNA helicase and DNA ligase, terminal deoxynucleotide transferase and DNA polymerase, AID and RAG1, and RNase and reverse transcriptase. So the correct option is answer number 1. That means recombinase 1 and the recombinase 2 are the important enzymes during B cell maturation for the VDJ recombination. Okay. Moving on to the next question, which T cell subtype plays a significant role in inflammation and is also linked to the autoimmune disease promotion? The options are Th1, Th9, Th17, Th22, and Treg. So the correct answer is Th17. So it is linked to inflammation as well as the autoimmune diseases. Okay, moving on. So this is the different functions of the T cells that are shown here. So we have the Th cells that secretes that have the CD45RA and CCR7 that secretes IL-2. Th22 is linked to the mucosal immune response, inflammatory disease, and barrier defense. Th17 induce inflammation, promote autoimmune disease. Th1 intracellular pathogen elimination. Th2 induces strong antibody responses and the allergic responses. Treg, maintenance of the cell tolerance. Th9, that humoral immunity through B cell interaction and functions through mast cell and other CD4 positive T cells. And TFSH, then it helps in antigen specific B cell immunity. Okay. Let's move on to the next question. Which term describes a viral population within a host that constitutes of many genetically distinct variants die to a high mutation rate? So what can be the correct answer? So it's quasi-species. That means that a viral population that have many distinct variants that die due to high mutation. Okay. Moving on to the next question, heptanes require a large carrier protein to elicit an immune response because they are too large to be recognized by the immune cells on their own? No. They can trigger an immune response independently? No. They are too small to be immunogenic themselves? Yes, this is the correct answer. And heptanes only react with innate immunity? No. They are only recognized by T cells? No. So the correct answer is option number 3. Okay, let's move on to the next question. What is the significance of kappa and lambda light chains in the antibodies? So they are responsible for the flexibility of the antibody structure? No. They determine the antibody isotypes? No. Okay, isotypes are determined by the heavy chain. So the constant region of the heavy chain. Then the kappa and the lambda chain contribute to the genetic diversity in immune responses with kappa predominant in humans. Yes, this is correct. They allow antibodies to switch between IgM and IgG. No, they are involved in the complement activation pathway. So the correct answer is option number 3. Okay, so they help in increasing the genetic diversity in immune responses. Okay, let's move on to the next question. When is IgM produced during an immune response and what is its primary function? So the options are IgM is the first antibody produced during secondary immune response, primarily involved in placental transfer. No, this is the function of the IgG. IgM is produced later during an immune response and is responsible for allergic reaction. No, this is for IgE. IgM is involved in autoimmune diseases. No. IgM is produced solely for parasitic infection. No, this is also for IgE. It is the first antibody produced during an primary immune response and is essential for early immune response. Yes, this is correct. So the correct answer is option number 5. Okay, so IgM is the primary antibody that is produced and it is for the early immune defense that takes place. Okay, in the structure of the antibodies, which complement is directly responsible for antigen binding? So the correct option is the complementarity determining region, that is CDR. This is present in the FAB region, so that determines the antigen type. Okay, so correct answer is option number 3. Let's move on to the next question. Which of the following statements about antigens and immunogens are correct? So all antigens are immunogens. Uh, I think no. So all immunogens can be antigens, but all antigens cannot be immunogens. Okay. Then immunogens can induce an immune response, but not all the antigens can. Yes, this is correct. All antigens are always immunogenic. No. Okay. Immunogens are always recognized by the immune system. Yes. Antigens may or may not trigger an immune response. Yes. Immunogens are involved in triggering antibody response, antibody production. Yes, technically. Antigens are always harmful to the host. No. Only immunogen can be used in vaccine development. Technically, this is wrong. Okay, so the correct statements are 2, 4, 5, 6, and 8. Okay, so 8 can be correct but can be wrong because the immunogens are not the only one that is used in the vaccine development. Although they elicit an immune response, so it can be considered. Okay, so let's move on to the next question. Which of the following statements are correct? So BSA is a poor immunogen. No, it's a good immunogen. Okay, that have a 69 kilodalton size and it will be immunogenic in humans. Then poor immunogen size will be greater than 10,000 daltons. No, this is also false. Serum specific antigens are immunogenic in nature. Yeah, this is correct. Then B cell epitopes tend to be on the outside of the antigen. Yes, this is correct. Upon immunization with glucagon hormone, B cell and T cell epitopes won't differ. No, this is false. Then monoclonal antibodies are used in pathogen detection applications. Yes, this is correct. The antigen recognition of TCR is weaker than BCR. Yes, this is also correct. TCR binding is strong as compared to antibodies. No. Complex cellular assays were required to reveal the presence of the TCR. Yes, this is also correct. So the correct answers are 3, 4, 6, 7, and 9. Okay, so let's move on to the next question. Serum antibody titer is determined by which of the following methods in the hybridoma technology? So it is flow cytometry and ELISA. Okay, so the flow cytometry will detect the number of antibodies that are produced. So they will detect the flow in the flow. In ELISA, the concentration of the antibodies will be detected based on the absorbance or fluorescence. Okay, the MTT assay basically determines the viability, and Western blotting will not be able to use for the detection of basically the antibody titer. Okay, so the correct answer are 1 and 3. Okay, so this was a repeated question. So this was the last question for today. So we'll close the session for today. Thank you guys for joining in.